And without further ado, it's my pleasure to introduce our first speaker, Frank Von Delft from Diamond Light Source and Oxford Center for Medicine Discovery, who'll be telling us about moonshot lessons for open science. And with that, I will let you share your screen. I think you have to unmute, Frank.
Good point. Thank you. I think I'm looking at the right camera. Yeah, this is quite an exciting initiative. I must say it's John's idea, and I think it's brilliant that we have a way to communicate with people that think about open science. And because the ASAP project that is funding us is really structured around open science, it feels right that we should be opening the conversation. So I'm going to put down some lessons that we learned through the COVID moonshot, which I'll briefly introduce. And that are at least affecting some of what we need to do in the ASAP project. So I come up with a rather lengthy title: Moonshot Lessons for Open Science, Sustaining a Collaboration Platform for Real-Time Data. Blah. I think maybe I should have shrunk it a bit. How data infrastructure should accelerate open science, and so I'll try and just share lessons. They may be a little bit disjointed because it's actually not that easy a problem, but I think we've learned quite a lot of interesting things along the way. So as always, I should acknowledge a large number of people that have been involved with certainly the XChem project and over the years, and in particular because the focus here is the COVID moonshot and ASAP as well. But for these slides, thanks especially to Darren and Warren, who gave me some last minute help on some thoughts, which I'm trying to crystallize these things which are a bit diffuse. And so as you probably will notice, and obviously the COVID moonshot itself was a huge collaboration, and you can go find them all on this URL later on, but it was quite an exciting task. So straight to the topic here, when we talk about open data, I guess most people would think, well, the best I can achieve, thanks very much, is I do my experiments and try and at least somehow zip up my data and shove it somewhere where people can find it. And that certainly is not that easy to do, but it may not be enough as we go into the next decades of doing science. We probably have to think a bit harder about it. And that's, I think, where we can try and do something foundational and be much more alert. If anybody needs introduction to what XChem is, it's basically fragment screening and crystal structures. It's a facility we built up at Diamond Light Source over the years of the last decade or so. It's supporting what is one of the, if you like, gold standard experiments for fragment screening. It's quite a tough experiment to do, so we set out to streamline it here at Diamond Light Source. And we have quite a pretty large user community. We've been running for a number of years now and have generated a pile of data, some of it open and much of it else archived here. And there are similar initiatives around the world, but essentially this operation had been running for some time when the pandemic, when the coronavirus came by and helped us all focus the minds a bit. And what happened in the pandemic, so this is really skipping over a huge amount of work, but just before the lockdown even started in the UK, my colleague Martin Walsh's group managed to get the main protease crystallized in APO state, and it's some magically fast work. And then this is you can find this in a Nature Communications paper. Basically, these lines here show how fast we managed to move back at the start of the pandemic and get a bunch of structures in the public domain, because that's what people were doing at the time. And the whole thing snowballed thanks to some thinking from near London and also Alpha Lee and John. And then before we knew it, we were sitting with this big spontaneous international collaboration and trying to get see if we can convert these fragment crystal structures or crystal structure fragments in the main protease into an actual drug. Somewhat out of desperation, but also maybe building on some prejudices we had about how science might run. And this thing, this whole project is still running, going strongly. This is not the time to give you an update, but it's currently in the development phase and funded by the Wellcome Trust at this point. What I want to point out is that here in these phases of fragment to lead, which we managed to do in about 6 months, and then in the well, you can call it lead optimization stage, what we were doing from Diamond's side for the project was generating a very large amount of data to try and help progress these things as fast as possible. We were generating 3D data, and that was our contribution. And I think Darren Fearon actually did the bulk of this work throughout the time. Somehow, I don't know how he did it, but he was generating structural data for the moonshot. And here's a timeline looking over the weeks of the projects, and these dumps here are the number of compounds that were put into crystals as it went by, and you can see every time the shipments showed up, a whole bunch of data sets were generated. Sorry, the blue are the data sets and the orange are the compounds that were provided to the project that were sent to Diamond. And over time, you can see the number of diffraction data sets increased to a very large number by any standard, to be honest, for such a discovery project. And the number of compounds that were added was maybe these were done in triplicate, approximately, and about 40% of these became crystal structures, which Darren had to somehow get out to the collaborators. And the goal was to get as fast as possible turnaround to the design team, and if at all possible, to try and get these data available alongside the assay data. I think as a pragmatic thing, because this was without a whole lot of infrastructure that we really need, he managed to at least inform the most important data points. And not all the data has actually made it out yet, because it's a huge pile of curation, which I'll come to in a minute. But broadly, we were trying to, or Darren especially, was trying to keep up with the development of the compounds. So here is this is really the context of what we learned. A lot of other things happened as well during the pandemic at XChem. Up here is the timeline of the number of infections in the UK. Of course, at the beginning, there was very little testing, and later we had peaks, and the red lines are the lockdowns. Initially, the user program was suspended because we always have users coming here, but it was a number of COVID-2 projects were still being run with collaborators, and at the end we did about eight of these fragment screens against different targets, and those data are actually publicly available and have been published as well. So a lot was happening at this time, and we're trying to get the data out as possible. And initially what we had to do for the start of the moonshot, well, what triggered was we essentially stuffed it on a web page. It's like a pre-print by web page, if you like. Not particularly elegant. It was a bit of a scramble, obviously, and the content you can put out there is rather minimal. But it was there, and there were these links for downloading the zip files of structures. They didn't even go into PDB because that itself is a lot of work. And then this set the model for some subsequent releases. This one is the project we did in collaboration with Jamie Fraser. And so essentially, we had this rather unsatisfactory way of putting the data out by sticking it in a zip file somewhere. But as we simultaneously with that development, we'd actually got this platform called Fragalysis mature enough to also make it useful in the project. And I'll say a bit more about Fragalysis in a minute, but it broadly, this crystallizes the problem we have, that you do these 3D structures and they're all in crystals, which is essentially an artifact. So you have to do something to that artifactual data to maybe get it into a common reference frame so that people can just, the ambition is that you could just see the data. You need to make sure that these things are consistent. You know, if you've got side chains that are moving around and things are disordered, you need to try and somehow get it in a somewhat uniform format. And this is non-trivial at all. And then you've got this pile of structures, you know, dozens of them if you're unlucky. And you start getting quite uneasy about putting them out. So ideally, you've got some infrastructure to help others to take a look at it. But you know, you can send them from the files, but it becomes quite disjointed. So we did build some prototype review and software where people could take a look somewhere else and could send some comments back to the refiner and hopefully get a better quality out. And this was all cobbled together, I should say. So a lot of it's prototyping. And then once it's generated, you ideally would like to annotate these data because you might have seen something about them. So can you say something about the ligand quality, or maybe this annotate the series? All this is prototype, I should say, but it was quite clear that these were needed if you're going to make have the data make sense. And then you shove it out into this platform, Fragalysis platform, and possibly beyond that go to the repositories. But there's another complexity step of putting in the PDB. So Fragalysis was our primary sharing platform. It looks, well, it's a 3D viewer basically, but then it's got some navigation tool for the structures. You can go find this stuff there if you haven't yet. It's basically, in the first instance, it's meant as a collaboration tool for 3D data, sort of ligand-centric. So you want to see the ligands that are of interest. It does support uploading predictions or computational data as well. You can look at them in this case. We've structured it so you see on the right-hand side. But the idea is that your view of the ligand or the ligands should be that binding site of interest, and that you don't distract users by, well, that users can simply go and navigate to this information and start making sense of it, hopefully. It's sort of scientifically in a sort of alpha beta stage, but it is a well-engineered cloud stack, so people could deploy it if they want to. There's warts and all. And there's a load of open data uploaded onto this, which you can find. These were generated through the SGC projects and through COVID, and for some definition of fair, you could consider it fair. A lot of it was funded by Janssen, I should add, through the IMI project, and we have contractors helping us develop this. So this platform has been taking a lot of our time to see what we can do with it, and it's also a key point in the structural side for the ASAP project. So broadly, the goal of it is to support sharing and analysis. Now, if you think of the data that goes in, as I just discussed, you have to review your data and get it ready for release. And I'll come back to these complexities in a minute. But then once it's on the platform, what we're trying to support is this idea of sharing a view of the data. So we have this concept of a snapshot, which is if you're seeing something interesting, you can just click up there and say, share, and it will generate the URL that you can send to your colleagues and say, look at this interesting thing I saw. And you can send it by email or stick it in Slack or anything, and then that view becomes an entity and it actually persists. So, well, as we developed, some of them broke as we went, but that's we're building this airplane as we were flying. At the same time, you can also download data. Initially, you had to pull the full 5 GB stack, and then later we managed to make it much more selective. And also what we're working on now is that you can push these data into certain computations to try and pin down the analysis of them. And these could be an analysis computations, but they could also be enumerations or further progression computations. The framework is quite broad, but broadly, the point being that the computation of the data is part of understanding it. And it's one thing to just shove data out to the world, but you probably have to work harder if you want, like, people that aren't actually computational geeks to make sense of it. And so these are 3 destinations or outputs of the data in the platform. And this is I'll come back to this. What do you do with this data and what are the complexities? But I think this is maybe the nub of open data version 1 or 2, not version 0. Or you could say maybe the dumping into repositories version 0 open data, the sort of being able to guide the user to the view of the data that matters is maybe version 1, and then allowing further annotation is where things get more complex. Now I'll just elaborate this. But these snapshots actually were quite an interesting feature early on, about 2017, when we were first starting to try and get this platform ready. We suddenly lit on the idea of this, as I said, a persistent URL for certain views, and that became mature in the course of 2020, because we tried to push the feature and make sure it was more or less stable. As I said, it's warts and all, but it's there. And then once we submitted preparing the submission of the ASAP grant, or actually the post-submission, we thought, well, maybe we should just look at those snapshots. They're all in the database, and we can look at how many have been generated over time. Use a timeline, and these are the number of snapshots in there, and then we can characterize them by the different targets that were being looked at by various people. And you can see, obviously, it's dominated by the main protease, because that was the active moonshot. But there were some other things that were happening. For instance, there was the MAC1, so the macrodomain paper, which Jamie probably will talk about now. That was accepted at this point, and so you can look at what happens here. Well, sorry, here was a jump. So we tried to put some things we knew about onto these little jumps here. So one thing was that at Imperial College, they had a master's degree. I know this because the person who ran it said, oh, this Fragalysis tool is so interesting. we structured our course around it. So I went back and asked her exactly when they did the course. It was exactly in this time. So there were these students playing with the data and making sense of it and clearly generating snapshots as they went, which is kind of cool. And you can look at some other of these bumps. That's usually when new MPro data was released, and that obviously then people pile onto it and generate snapshots and share them around, so that you have little jumps. When the Mac 1 paper was accepted, that also was in Fragalysis, and there seemed to be quite a lot of interest at that point. We didn't track exactly who was generating it. And here's a little other thing when the NSP16 data was released, which is that light green one. I just blew it up here. We told this non-affiliated pharma group that was interested in it, they had been bugging us for months with the data were ready, and it was a struggle to get it ready for technical reasons. The moment it went out, bang, you can see there, people apparently started looking at it and generated a number of snapshots. So it seems from this that this kind of way of presenting data, not just putting the data out, but allowing people to share views of the data is useful and in demand, at least by this evidence. And certainly what I was very intrigued to see these conversations on Slack, sort of always sharing snapshots and people complaining when they broke, because that happened as well. So that's reassuring. The other thing that happened was, as I said, the downloads. So people like John Kadira and this group who were trying to do this modeling on the folding at home, well, they would like to get the data, and the data which needed to go up. So rather than initially it was just you click the button and you get this huge big zip file, which was, as I said, maybe it got bigger and bigger. And so we then refactored the whole thing to allow people to download a snapshot of the data based on what they were seeing on the interface. And so you give people more control over that, again, just based on the interaction with the data, not having to go and do some coding in the background. So you could, for instance, if you see a bunch of structures on 3D display, in principle, you can get those downloaded, or maybe on the hit navigator, that's this other tool on the left, you can select things over there and get them downloaded, etc. So you can define the snapshot. But what was more interesting is that what we tried to implement, and I think it works, as far as we know, it's stable, that once you download, it creates a snapshot in the database of that download. So you can get a permalink of the download at the time when you did it. And that can either be preserved, so you get the snapshot of that current status, which never changes, or you can have an incremental download. So it says, this view I had alongside any other data that came as you go, bring it down, download it. So giving some flexibility, and jury's still out if this is flexible enough or too flexible or pointless, but there's another point that the download is self-documenting. So we went to some trouble to make sure the PDF would describe what was in that zip file, because that's the other thing, when people download files and send them around. So, but the broad challenge is, this is a technical approach. The question is, if you put data out there, how do you allow people to both use it in their own tools, but still have a presence online that it can be shared? And that's sort of the ambition here, that this permalink should be able to work. And also, once you've got the download, that download itself has a snapshot, one of these ones, which you can then go and look at. So within this README file, this permalink is stuck into there, so you can always go find it again, navigate back to that view of your data. So it's a funny problem, right? You've got this platform with data on it. You allow people a view into it, then you allow them to extract data from the view and go off and do stuff. How do you allow them to still get back to the view that generated the data? So that's what we're trying to solve here. I don't think it's an easy one. I'm sure there's many ways to break this, but we try to think through it. So that's the scope of the problem. That's a technical solution. We're trying to work through this more and get it stable in the course of ASAP, so that really our 3D structural data can be brought out and made persistent for communities, because we will be generating a very large amount of it. So I'm now going to go off into a bit more speculative things, or think a bit harder, more about the shape of the problem of putting data out there, and what are the problems that have to be solved. So here are 4 projects of three projects of our COVID work. The first one is the main protease, and as you know, we put the data out very fast, and then we managed to get a preprint, and it took quite a long time and a lot of stress to get the thing actually published. But maybe this is reflection, not that slow. Then the macrodomain paper, that took much more work. It took longer because, well, people were trying to just get by, but eventually it came out maybe just a year afterwards. And then some other, the next screen, every time it goes a bit longer. There's a few other data points we didn't get around to putting down, but basically the time between, the time between the experiment and the thing actually being formally published in a formal publication gets longer and longer. And so there's an interesting question about the psychology of publication. So I'd say, for instance, here's a case. So we put our data out in a big rush, of course, and so you give it a lot of visibility, and people obviously go poke around at it when they have time, and that's useful and it's important. And then, well, this is one example where there was even a commentary published in a peer-reviewed journal about the use of that data. And actually, some of the conclusions there are quite strong. They said, well, these data are dirty and are polluting the PDB, and that's a viewpoint. I respect it, but it was articulated. And so the one reaction would be eye roll, like, come on, this data like triggered the moonshot and it triggered ASAP, and the data was used explicitly, cited by the Shinogi project as having been useful for triggering stuff. And so are we really going to get parochial about the quality of the data, which is what this paper was commenting on? But we have to think harder about it, because it's making an important point that we need to think about the shape of the paper, of the data that we put out, and what can be done with it. And I think this is our challenge in the next decade, if you like. An hour, I'm talking in the broad sense of the structural biology community. Because there's also the psychology around publication. Think about why journals can still charge us a lot of money. So, I mean, some of the polemics that go around is, we academics are fools because we just let ourselves be charged. But maybe we are getting some other value out of the journals, which is that we're scared to put stuff out there. We'd like to know that there's a gatekeeper that is helping us not make a fool of ourselves with our data and our conclusions. And so that has value, and people will be willing to pay. And I suspect we're sitting in between this. So the more bold people will say, I'll put my data out, but if you're not as bold, then it's maybe quite comforting to know that, well, there's a reviewer and an editor, and the thing will be looked after. And that is value that the academic community, I think, will stay willing to cough up for a while. The other interesting thing is, why did the preprints shoot up in the pandemic? Is it because urgency finally trumped this fear? That people then started saying, oh, right, that's, we need to put it out, never mind the quality. So I've been involved in data release, open data release for many years in multiple projects, and I've always seen how scary it is for people that to push the button to say this data is now going live. So I think there's an important psychological point, and you can instantly put the data out and suddenly you're part of Twitter mob. So I think one shouldn't underestimate that. So the platform ideally would help with that. And here's the sort of problems we have to then deal with. So you can put the data out, and as I said, we've got these entities that consume the data and make them persistent. So initially we've got snapshots and downloads, latest coming computations, which you can explore. So what happens? You put the data out, and then people do stuff with it, and then you put more data out, and maybe this data is revisions and deletions, and because you've looked harder, or new observations of the same experiment. So what now? What do you do with these persistent objects? And then you generate more persistence, and more data comes out. So you have this problem of interplay between the two, and you have to give some way to give visibility to things that have changed. So it's not actually that easy to work out. And so the other thing that struck me is that we may be stuck in like 40 years in structural biology, maybe stuck 40 years ago. Because if you think what happened to looking at 2D plots, if these are your data, back then you do single plots, and people say, come on, you have to put error bars, and everybody gets whipped to do error bars. And now you get whipped to, if you do error bars, you get told that's rubbish, you should put violent plots or activated violent plots. The data are much richer than just a stupid little plot with a whisker on it. So we are evolving, but I don't think structural biology is evolving, because right now we are generating multiple views of the same experiment repeatedly and easily. The compound gets shipped again, and we've had this in the moonshot. So you have on the same compound 3 views, and what do you do with that? Do you confront the medicinal chemist with all 3, or they might just say, sorry, I just want one. What's the truth? Well, I don't know what's the truth. So do I annotate it, or so how do we give this? We have this other concept we've been developing that one can annotate atoms if they're not very well defined. So you've got this bad atom idea. It's a kind of error bar, if you like. So we're trying to visually communicate that alongside all the other views. So I think we've got a lot of work to do. If you start adding simulations into this mix, then it gets even more complicated. So I think we're, as I said, 40 years old in terms of representing 3D data, and we have to think much harder about what to do with it. I've got 2 more slides just to put some polemical, or let's say prospective questions out there. What about discoverability? You know, we'll be generating a lot of structural data, moonshot times 20. So it's a pile of data that goes out there. I mean, how can we let people know what's there? And I mean, these are PDB files. You can't discover those. So can we, for instance, make these snapshots Googleable somehow? You to give an index and keywords and stuff. So, or do we need to rely on Web 2 technology? You know, the whole social media approach. Like, what is it that maximizes eyeball or gets maximum outrage? So we introduce errors to get the profile of our data up. Who knows? But I think we have a problem, and we have to tap into what's been explored in that space. Or maybe we just let people go through collaboration networks and that's enough. But I suspect it's more interesting than that, about how we can actually find a way to index and discover the data and something. And then on top of that, so can we make the data better through annotation and exploration? And by this, I mean, when people look at it, can you allow them to, on the platform, make comments or run analyses and then comment on those, or have discussions about those analyses, so that, and that's all captured as part of the data. So it's not metadata, it's more enriching things. So allowing on-platform analysis, capture of conclusions, sign off of conclusions. So someone else says, yeah, I agree with that conclusion. I put my name and my date onto it. And you attach them maybe to certain atoms in a certain part of the, you know, part of the space. So it's a complex problem, but I think that infrastructure is worth exploring. And so a final word is, I saw this blog post, I was hoping to find it, but somebody from Microsoft was making this point that if you look at our current AI, which is what's being done in all sorts of web content, you've got these things of Mechanical Turk, etc., where you have loads of people all over the third world actually trying to make money by doing very boring clicking on images and stuff. And so it's essentially mistreating the annotators as low-skilled workers. And the point they were making is that generates poor data, and that will generate poor AI. But if you instead embrace your annotators and allow them to sort of engage with the engines that will learn from the annotations, that will allow you to get much closer to what you might call good AI. So AI that's intelligently fed with data. So you need to have your annotators as part of your ecosystem of learning. So you need to sort of have a way to rapidly assimilate these annotations, make the implications of the annotations visible to the annotators so they can understand what it is that they are doing to the machine learning model. So they need to be part of it. So allow them to interrogate the models that are generated by the annotation, give them agency to edit these things. So you really think of them as an ecosystem. And so the big question is, for the sort of data that we're generating in sciences, should we be supporting that and working out how our infrastructure can support that? It's a big ask, but I do think this is where the next 10 years will be going in terms of machine learning. That's the end of my verb.
Thank you so much, Frank. That's an exciting exploration of an amazing data generating resource and the challenges in making that data connecting with the target audience who's doing awesome science with it. So I didn't see any Q&A questions. We're a little bit over time.